As opposed to Juliet and the other researchers here, my background isn't academic. It's 30 years of doing risk assessments in the field. So initially, risk assessment of microplastics is presented as an intractable challenge. Is it really possible? Well, yes, it is possible. And we can apply the standard chemical risk assessment paradigm here, that risk is a function of hazard and exposure. If there's no exposure, there's no risk. We can apply this logic later, as we'll see. So microplastics present their unique risks in terms of extreme diversity in shape, size, and composition. It's true that currently we don't have enough data to quantify the hazards of microplastics, but we can make conservative assumptions on their toxicity based on their polymer composition. The case in point here is polyamide. We know that polyamide is a constituent of most house dusts. We know that polyamide is especially toxic to lung tissue. We know that polyamide can leach toxic contaminants like heavy metals, chromates. And we know that polyamide particles can particularly form nanoparticles. They're quite prone to that. So we would, on this basis, it's perfectly reasonable to assume that polyamide particles or a house dust represents a high hazard category, or a high hazard category, yes. Regarding exposure, we can reasonably quantify external dose, systemic exposure, and internal kinetics using modeling techniques based on particle numbers, size distributions, and shape. Now, we don't have a lot of these data already, but we're implementing a framework that can take in these data as they become available. Until that time, it's perfectly reasonable to have an exposure-led risk assessment, considering the special vulnerability of early life stages. With that in mind, we have developed a framework. A framework is essentially an infrastructure that allows for building over time as new data and better data become available. The framework is modular and extensible, currently tailored to early life stages, but can be extended to other scenarios. We've developed a conceptual or aspirational database that lays out exactly the data we need in order to do a quantitative risk assessment. We've developed interactive exposure scenarios for early life stages, and these are available as web-based models. All parameters in these models are empirically anchored. That's an important point that I'll come to again later. And on top of this framework, we have a phased roadmap that takes us from what we know today or what we don't know towards a fully quantitative risk assessment in future once the data are available. So as a first step for hazard characterization, as I discussed for polyamides, we take a conservative hazard banding approach. We have a scoring system in our models for hazards, and this is based on a publication from the AURORA project 2 years ago that lays out, based on polymer composition, a broad hazard banding for particles. And that's a perfectly reasonable approach when you don't have quantitative data. We've also built a particle database. Again, this is the aspirational database, and this provides a central repository for microplastic data. On top of this database, we have an exposure matrix database. What this does is it takes in the different particles, so you can define reference exposure media, for example, a house dust or a sample of food or water, and that allows you then to bring that directly into the model, that you can use that as the basis for your exposure composition. You can basically provide a composition for the entire exposure scenario, not just one particle and not just one exposure medium. Once the data in this database has become available, we will be able to do things like model cellular interactions, model agglomeration and deglomeration in physiological fluids, model the protein corona effect or the strength of protein attachment, and also Trojan horse effects, that's contaminants that may attach to particles of different size. The exposure models are built on 4 proof of concept scenarios. Each of these models addresses unique exposure pathways per life stage. The first scenario addresses maternal and fetal exposure. So this is a maternal exposure from the outside, the external dose. We can model how particles can move into the systemic system and then into the fetus via the placenta and placental translocation. We have a neonate model, which really looks at the feeding risk from using polypropylene bottles and particle shedding from polypropylene bottles. But we also model the crib environment at resting breathing rates. Regarding bottle shedding from polypropylene, the data we have recently suggests that with repeated heating and sterilization cycles, that the plastic shedding from these bottles actually increases over time. So that in itself is already an actionable risk mitigation strategy that we'll talk about in a moment. The third scenario regards infants crawling behavior, dust resuspension from carpets, for example, and also hand-to-mouth behaviors. The fourth scenario looks at toddlers. This is a dual pathway model. So first of all, we model particulate exposure from dust inhalation and ingestion, but we have also provided a model for chemical leaching from plastics. Now, the thing here is we don't have good data for particle shedding from plastics that may be mouthed by young children. You know, children put toys in their mouths. However, we can model the leaching of chemicals, and we have reasonably good data from this in the past. So although we're in a data poor situation, when the data become available from plastic shedding from mouthed toys, we have already in place a placeholder, a methodological placeholder that we can plug our data into. So very briefly, how do the models work? Well, there's a web page where we pull in the exposure context from the database that I mentioned. So here we have inhalation from house dust with drinking water matrix and a dietary matrix. This pulls in all of the data from the particle database. So we can model the entire exposure context. The models then run Monte Carlo probabilistic simulations on these data and the data ranges. So the data ranges that are provided by the user include, quantity of water drank per day, food eaten per day, the dustiness of a particular room. The distributions or the exposure distributions are provided in terms of the external maternal dose, a systemic dose, and the final fetal dose. So this is possible because we have kinetic lookup tables that can provide translocation data. The models are fully particle size and morphology aware, and this is an innovation. We can provide the particle size distribution of an entire exposure scenario, not just a single particle or a sample of dust, but everything, including water, food, dust exposure, and any other media we want to define. And from this, we can do extremely detailed modeling of internal kinetics, translocation. An important point here is for policy work, we have, does this still work? No, it doesn't. We have prioritized pathways. So we can map out for every exposure scenario that a particular pathway is more prominent, and that already points to policy actions that can be taken. Yes, results, we see the hazard score here, but also the results are presented in a 3x3 risk matrix that already gives an overall impression of the risk posed in a particular scenario. So what we have is a scalable risk assessment framework supporting the work of the AURORA research partners. As we've seen, we apply exposure models that already derive actionable risk assessment or risk mitigation strategies. You could say what we're doing is we're providing a scientific basis for what most people already see as common sense actions around keeping your house clean from dust, using wet dusting techniques, vacuuming, very simple stuff. Our framework delivers an infrastructure for standardized microplastic data sets. It incentivizes high quality data generation. If you don't agree with the models or you think the model is too conservative, fine. Here's the data we've defined in our database. If you provide those data, it's already plugged into the models. We can run a more precise quantitative assessment. The models enforce scientific transparency. Everything must be anchored in empirical data, and that confronts uncertainty so that we know exactly the uncertainties. Any expert using these models know what we know and what we don't know. The framework also is overlain with a roadmap that charts us towards a fully quantitative and cumulative or chronic exposure risk assessment. So the question here is, if a particle goes into a fetus, is that particle still present when that person reaches adulthood? And finally, at the moment and in future, the framework offers a clear strategic direction for researchers, i.e. we provide what data we need in order to do a qualitative or quantitative risk assessment, and also for policymakers in terms of actionable steps that can be taken today to mitigate the risks from microparticles or microplastics. And that's it. So the models are built and the engine is running. We just need to plug in the data and start tuning, testing, etc.